Our next presentation will be by Dr. Andy King from the U.S. Meat Animal Research Center. Dr. King has a couple of degrees from Texas A&M University, a master's degree from Kansas State University. And I did look up his official title because I knew I didn't get it right a while ago. So research food technologist. And he's going to kind of follow up with some of these technologies and kind of been the theme of this entire program and really update us on some meat quality attributes with technology. Dr. King.
Thank you, Dr. Perkins. It's a pleasure to be here today, and it's a great pleasure to be on the program with one of my mentors. And I just want to do the caveat, I am a meat scientist. I sometimes pretend to be a muscle biologist. I am not a machine learner. So a lot of what I talk about machine learning will be fairly rudimentary because of Dr. Sable taught me a lot of things. He did not teach me about machine learning, but he did teach me two things that I think are really germane to my talk today. And the first one is that the best analysis to do with data is the one that tells the story. And so as we evolve and we get into some different situations, what we've been able to do or needed to do to analyze data has changed. The second thing he did is he taught a class called Carcass Composition and Quality. And as a graduate student, we called that the mystery hour because you never really knew where it was going to go. But one of the things that Dr. Sable did in that class was talk about some of the classic papers in meat science and talk about the context of what was going on at the time and kind of how that wound up being the publication that we've all read. And so with that, I'm just going to jump in. The approach I took to it was to talk a little bit about how the thought process of data analysis has changed with some of the technologies coming along, and then kind of wind up at a technology that we've been involved with a little bit that I think has some promise in the meat industry. So one of the things that Dr. Sable talked to us about is he told us a lot about a man named Charlie Murphy, who was a big part of developing the grade standards in the US and specifically the yield grade equation. And so one of the things there's a lot of talk these days about maybe trying to redo the yield grade equation. I'm not going to get into that. I have some opinions, but that's not why I'm here. But I would like to say two things about this. One is if anything I'm working on right now is still being talked about 60 years from now, I'm going to consider that a win. And then the second thing is when this was developed, the approach to doing these kind of experiments is they went out and they had a list of maybe 10 or 15 variables that they thought might be related to carcass yield that they could measure. But they kind of at least knew or really suspected what those relationships were before they went in and tried to do this. And they developed a really nice yield grade equation, and then to predict yield. And then they kind of tweaked that to create the grades that we use in the US today. So if we're going to redesign this today, we've got a lot of technologies that weren't available then. And one of them is the grading camera that my group had a big role in developing and bringing into the industry. It's used to grade a lot of cattle. And that instrument takes an image of the ribeye at the 12/13 rib interface. That's all it sees, but just that cross section, they get somewhere in the neighborhood of 20,000 variables out of that. And most of those, if we even know really what they are, we have zero idea how that may be related to yield, how those are. And so we have very complex data sets. And so then we're in there looking for relationships that we have no idea whether they're there or not, what they might be. And so machine learning, artificial intelligence, neural networks, there's a lot of names for it, bioinformatics, and people who are really involved in the science of the math will quibble about what is qualified as what, but really these are using computers and technology to take really complex data sets and extract the information out of that and help that data tell the story. So with that, some things that are really going on in the industry that are really interesting to me as a meat scientist, the plants out there and feeders and even cow-calf guys are using machine learning and AI for process optimization. So they're getting all the data that they can get and they're feeding that into a system that maybe gets them a little bit closer to that fat content of the ground beef, or it maybe makes things flow a little bit smoother going through the plant. The other one that's really, you hear a lot about and you've heard about for a while, but it's really serious now, is automation. 10 years ago, you went into a plant and you started talking about, it wasn't unusual to hear the phrase, labor is cheap. You don't hear that anymore. And so usually we had to slow our line down to about 10% of what it normally is just to be able to run. So anything they're able to do on automation wise, one thing I think is really interesting is you used to see a lot of automated splitting saws in pork plants, and they would say you didn't see that in a beef plant because there was too much variation in size and those robots couldn't run, but they're putting them in beef plants now. So that has really come a long ways. The other thing, Dr. Sable and I were at the Reciprocal Meat Conference last week, and we watched a presentation where a gentleman from Georgia Tech was showing a video that involved taking images of poultry carcasses and actually running an individual model on every carcass to be able to fabricate those carcasses with automation. And so if you're fabbing a beef carcass, it's pretty easy to learn where those breaks are supposed to be, but every one of those carcasses is a little bit different of how it goes into rigor. And so you really got to know what you're doing to try to pull those cuts out in a way that they look like they're supposed to look. So that's really something that's come a long way very quickly, and it's picking up speed. Value determination is another place, whether it's the grading camera, other technologies. I'm going to talk about that just a little bit in a minute. And then on the research and development side, and where I'm going to spend the bulk of the talk, is talking about how to understand the biology and how some of this data can be used to understand what's going on. And so, our group at the Meat Animal Research Center is really interested in impact and looking at anything that affects quality, and specifically beef tender or meat tenderness, color stability, and flavor. And we've kind of taken on a role. We want to address all of those at the same time whenever possible, because it's a holistic process, and it's more of a systems approach rather than an individual thing. So anytime that we're addressing any of this stuff, we really want to understand what's driving the animal to animal variation in these traits, and not necessarily just what that treatment did to the meat. And to do that, we have historically used what I like to call component traits, and that's a measure like sarcomere length or postmortem proteolysis to tenderness that we know directly impacts that phenotype. And we've had a lot of really good success with that over the years, but on the best data sets we've ever had, we're only explaining 30 to 40% of the variation. And so when I get called in for my performance review twice a year, every year, the first question Dr. Wheeler asked me is, what are we still missing? Why can't you explain more of this? And I kind of look back and say, but you can't explain it either. But but anyway, so this has been something we've looked at. And so one of the ways that we've approached this is through omics. And so this room, I think, is really familiar with genomics, which is kind of the blueprint of what's available, what could be produced by that muscle or by that animal. The transcriptome is the gene expression. So which one of those switches got turned on and off due to interacting with the environment, or the proteome, which is all the proteins that are in that organism, or the metabolome, which is a list of the whole set of metabolites as those proteins actually function. And so we know if we were able to measure all of those, we know what the blueprint was, we knew what got turned on, got turned off, we knew what machinery was built, and then we knew how that machinery is functioning. The problem with that is that's a lot of really big, very complicated data sets, and it's data that's really hard to get. So like I said, I'm going to give a couple of examples. of things we're doing to kind of handle some of these data. I tried to stick with things that, one, that I felt comfortable talking about, and two, things that I see really commonly in the industry. And the first one that you see a lot, and a lot of machine learners, there's a debate on whether this actually qualifies as machine learning or not. But I think it does help you find relationships and complex data as principal component analysis. And so what PCA does is it takes a set of data set and it accounts for all the variation in that data set, and it derives a totally new variable that explains as much of that data variation as it can. And then it takes what's left and creates an entirely new variable that explains as much of what's left as it can account for. And it does that until all the variation is accounted for. And then you can plot those. So those are totally independent variables. They're not correlated. So then you can start to plot those against each other and start to maybe hopefully see some relationships. And so that graph on the top right there is some beef flavor data. So, you know, if we go out to a steak tonight and you might say that tastes good or that tastes bad. Well, to me as a scientist, that doesn't help me a whole lot understand what I need to do. So we train people to break that data, that experience, that taste down into about 30 or 40 different variables that describe that. So there's stuff like beef flavor identity, fat-like, that are really good. And then you have stuff like putrid, spoiled, barnyard, that maybe you don't want your steak to taste like. So that's great. But if you analyze each one of those variables independently, you have descriptors of 30 different variables, and it's really hard to know what to do with that. So if you do the PCA, you can plot that and start to break that down into groups that kind of track together. And then you start to put together what's going on and what maybe can help you address consumers or processes to impact flavor in a positive way. And then the other thing you can do before down that is, so that's from a project we did that had a lot of dark cutters in it. And then I mapped all the metabolic data that we had on those samples to the flavor data, and you can start to see how some of those variables impact and relate to those flavor data. So you start to understand that, well, maybe, you know, glycolytic potential contributes to heated oil flavor or something like that. You start to understand how those things relate. So kind of an offshoot of that is clustering data. So if you have a data set that you have a lot of variation in, a lot of things going on, but you don't really know what to do with it, you can run what they call a clustering analysis. And so that starts with all the data, and it goes in and it finds the two that are most like. And then it finds one that's like that. And if it doesn't have a fourth one that doesn't quite fit, it'll split off and with a new wing. And so you can do that and you can decide, okay, I want to stop with 4 variables. And so this is a data set that we had an intern do a couple of years ago, where he went in and we took an in vitro system. So it's basically a test tube that had all the things in it that you needed to run glycolysis, which is the reaction that results in pH decline in postmortem muscle. And so Dr. Sable mentioned a lot of things that talked about increased carcass weight, increased carcass size, increased fatness. Well, all of those affect chilling, and which affects the biochemistry of what's going on in that muscle, which will impact final product quality. Unfortunately, we don't have a great handle on exactly what the control points ought to be during that chilling process. And so that's really what we're trying to get at here. So I had that student take that that in vitro system, and we mocked 2 different temperature decline rates. And so I had a bunch of data that didn't really relate to the treatments that project was part of, but I thought there's a lot of variation there. I thought it was really interesting. So we ran the cluster analysis, and what it found was 4 categories that broke up, that when you had a very rapid temperature decline, they ranked in that in vitro system a certain way. Just point out the red line at the top on the graph on the left. But if you did that through a slower temperature decline, those groups totally re-ranked, and that group became by far the fastest group in the session, and that were correlated to different things in our data set. And so right now, one of the big pushes in my lab is trying to understand what about the enzyme systems that are in that muscle that reacts differently to the different in temperature decline. And so that was something that we would not have found without a cluster analysis. So one other tool that we've been able to use for the metabolomics is to be able to use mass spectrometry. And so you can either use that to measure the proteome or the metabolome in the muscle, but it requires a lot of sample prep. It's very time consuming. It's very expensive. But you can run a sample through it and you'll get a series of peaks like that that tell you what the compounds that are in that muscle sample are. And you can run a library of standards through there and use that to map to that fingerprint to know exactly, okay, this sample has a lot of glucose in it. This sample has a lot of malate in it. The problem with that is annotation or identifying those compounds is really difficult right now. So that's something that hopefully will come along, but it generates a lot of data. And so I'm going to kind of walk through a study that we did a couple of years ago. So we went out to a plant and we screened select carcasses for tenderness differences. And we really were looking for extremely tender and extremely tough samples. We measured shear force, we measured proteolysis, and then we tag teamed with the proteomics and metabolomics lab at Colorado State and Keith Belk and his group to look at run those through a mass spec to get the samples. And actually the way I analyzed this data, it was very similar to what you would have done kind of several years ago for a genome-wide association study. So I ran an ANOVA, or analysis of variance, for every compound that we found, and corrected that for multiple testing. And then I used correlations to kind of limit down the variables that we wanted to look at in the data set. And then we ran partial least squares regression, which would be a machine learning technique to try to understand the relationship of those variables to tenderness. So this is just a measure of the differences we got in tenderness. And I showed this to a guy who's a VP at one of the large beef companies, and I thought he was going to have a heart attack until he understood that that didn't come from one of his plants. So because a mean shear force of 37 kilograms is tough. So but as we walked through there, we started out with a total of about 2,500 metabolites that were profiled. And out of those, 651 were actually related to our tenderness treatment at a very strongly corrected level. And then about 250 of those were at a nominal p-value for tenderness. And then we went and looked at the strength of the correlation to both slice shear force, Desmond degradation, and sarcomere length. And all said and done, we wound up with 102 that we put into our PLS regression. And so that's one of the things you got to do with these big data sets. You start with this big mound of data, and you've got to cull it down to the meaningful stuff. And so just to give you so unfortunately, out of that 102 metabolites, we only knew what 28 of them were. And this is kind of the list, but free amino acids related to postmortem degradation, a lot of glycolysis and Krebs, which is why we're interested in the pH decline and some things like that. Glycosides, and then fatty acids showed up as well. And so if we put all of that into partial least squares regression, which is an offshoot of PCA, which is why you have a similar plot, you start to see how these things all relate to tenderness. And we're actually right now, we're trying to use glucose 6-phosphate, and malate as a metabolic fingerprint to try to understand what the metabolic status of that muscle was and relate that to tenderness levels. So that's kind of an overview of how we use a mass spec for metabolomics. Like I said, it's a lot of sample prep. It's very slow. It's very expensive. And while we were involved in this, the group at Colorado State told us about a new technology that they were investigating at the time called REIMS. So REIMS stands for Rapid Evaporative Ionization Mass Spectrometry. And so now you know why we call it REIMS. So originally the REIMS system was developed for cancer research. And what it amounts to is it's a scalpel that heats up and cauterizes as it cuts. And so when you cauterize that tissue, it actually volatilizes some of the compounds that are in that tissue. And so they attach it to a mass spec. And so it will actually pull those volatiles into the mass spec, and it gives you a fingerprint a lot like the one that we got going at the really slow way. So one of the cool things about this is a very expensive machine, but once you have it, it's relatively cheap to run additional samples through. So it kind of removes some of the bottleneck that is involved with doing traditional mass spec work. The problem is as of right now, identifying those compounds is virtually impossible. Theoretically, you could run the libraries through it like you do for a normal mass spec and identify those. It's not quite that easy. So Dale Warner was at CSU at the time. He's at Texas Tech now, has one of these, and he's working on that. But what you can do with that is take those bins or the compounds that are come out of that mass spec, and you can develop regression equations that can help you predict a whole host of things. So Dale asked us to be a part of one of his students' PhD work. That student was Devin Gradell, and he did a really nice study where we went out and we went all over the place selecting carcasses to fit a number of categories. So we found some dark cutters. We found some top choice and prime, some low choice and selects. We went to Washington and got some Wagyu, and those are Angus Wagyu F1s, some grass-fed beef. We split the whole data set into tender and tough. And then we had the whole data set. After we did that, we did the ones that were from black cattle and the ones that were not from black cattle to see what REIMS could do to start to sort all of these things out. When all the data pre-processing was done, there were about 17,000 or 1,700 bins per sample, which is more or less the compounds that were in that mussel. And so then the other thing that Devin and Jessica Prinny did was to compare that to start using about 7 or 8 different machine learning approaches to see which one of those works best on this kind of data. So if we take our main data set, and so this is a plot, which is yet another offshoot of PCA, is partial least squares discriminant analysis. And so I will say, this is showing the first 2 components. Remember that this is the first 2. There's probably a couple hundred components that the PCA actually analyzed. And when you're looking at it and trying to analyze and interpret it, most of the time people look at the first 2, but there is a lot more variation explained. But in this data set, at least by the first 2, there was a lot of overlap, but in all of the quality grades, the only one that really stood out was Wagyu. And so there's a lot of things that are different about the biology and the growth curve of a Wagyu compared to a beef animal. So that actually makes quite a bit of sense. I'm not going to go into the different approaches, but you can tell that when you developed a model for all of these different machine learning approaches, there was a fair amount of difference in the accuracy of being able to split this big data set into these classes. And I'll talk a little bit more about the final model. So we also, Devin broke this up into a specialized data set, which was the Wagyu, the grass-fed, the dark cutters. And once again, you see that there's some differentiation in the data between those 3 different classes. And then if you run the prediction models, you have anywhere from 98% accuracy down to about 73% accuracy for the different machine learning approaches. The breed type data set, Angus or not Angus, or more accurately, black or not black. Once again, there's a lot of overlap in the middle, at least on these two, but there is a fair amount of separation there. And once again, there's multiple dimensions that the regression equation can pull from. And to predict there, we're right around 80% accuracy in being able to separate black and non-black. And if you think about how many breeds are black, that's pretty tall order, is to relate hide color to something. So that actually did really, really well, in my opinion. The tenderness data set, once again, a little more overlap than I was hoping for, being someone who really wants something to work really well for tenderness prediction. But once again, there's some overlap, but they did separate those out. And once again, about 90 down to about 80% accuracy in predicting those. And so finally, after all of that, Devin and Jessica went and took the best model out of all of those and developed the final model, and you can see there which approach actually did the best and how many variables actually wound up in the model. So you wound up with 24 principal components for one of them, or 60 mass bins, which is just a different way of kind of reducing that data. But your final accuracy went way up with those final models. So once again, I'm going to say that there's a good chance that these models are overfit, and overfitting means anytime you add a variable to a regression model, your prediction on your training set looks better. But if you overfit a model, when you go to a new data set, it's probably not going to perform very well. And so these were validated with a process called cross-validation. I'm not going to get into, but that really involves validating on the data set it was trained on. If we were to go apply this to a new model, it may not look quite this good, but I think it's really promising what this technology might be able to do. So in conclusion, the machine learning is a growing aspect in the meat science world. It's something that I never thought. And honestly, when Dr. Perkins asked me to do this and gave me this talk, my first response was, I'm not a machine learner. Are you sure you want me to do this? But I kind of, as I started to try to work through that and think of how to do this, realized that a lot of what we're doing in meat science, is machine learning. Now, I want to throw out the caveat, it's not at the level of some of what you've seen this week. And Brittany Keel works at the Meat Animal Research Center. She does some things that makes this look like really easy child's play, and it's really cool. And I've actually talked to Brittany about taking some of our data and taking it a little deeper, but we are able to use these techniques to be able to pull out the variation and be able to pull out the picture of what's going on in the data. So Dr. Salo, I think we're able to tell the story. So the machine learning is not going away. I just kind of want to bring that up. And I know there's a lot of concerns out there about machine learning, AI and all that, but anyone in here that happens to have one of these, you are heavily, heavily involved in machine learning. In fact, you're feeding them data on a minutely basis. So just, it's not as scary as you might think. Okay, I've kind of already covered the next one, but we're more and more, we're going to be looking for complex relationships. When I started at the Meat Animal Research Center, the director at the time, Mohammed Kamari, told me, I got bad news for you. All the easy problems have been solved. So, we're going to have to get pretty creative. The REIMS technology, I think, has a lot of potential. The problem is it's built for a operating room. And if you've ever been in a packing plant and an operating room, those two environments are nothing alike. So there's going to be a lot of work that's going to be required to make that robust enough of a machine to work in a plant environment. We need to be able to annotate more, but if we can annotate the REIMS, that really opens up a lot of possibilities on the research side as well. And then, like I said, I think we need to further develop models for robustness, but I think for where we're at right now, That's a really promising technology. And I'll just point out one more thing I kind of meant to say earlier, but I kind of left it out. It's not hard to find a technology that will predict tenderness. It's really hard to find a technology that won't falsely predict tenderness. And so it's more of a matter of teaching the model what not to jump on a lot more often than teaching the model what to jump on. And so with that, like I said, I have a great team, both support staff and collaborators that I work with all the time. I specifically want to thank the people in blue, Devin and Jessica, Dale, and Dr. Bell, for all of their work getting us involved with some of this. They were a big help. We learned a lot. And with that, I'll take any questions.